Yeah, just first, obviously, have to thank everybody for coming out this morning. And well, I guess it's a little later for most people, but uh, and of course, thank you for Michael for inviting me to give this talk. It's a great privilege to share my work with you guys. As I was introduced, I'm doing a postdoc right now in Steve Rohan's lab at the University of California, Berkeley. But I am originally Canadian. Most of the work I'm doing today was done at the latter end of my PhD in John Benziger's lab at the University of Ottawa, along with some of the work in the electron microscopy being done in Hugues Néry's lab at the Institut de Biologie Structurale in France. So we're going to talk about muscle type nicotinic acetylcholine receptors today. I was really looking forward to Radu coming in and introducing the family of pentameric ligand gated ion channels. But I'll give you a bit of an introduction to them. So the protein I'm studying, or I was studying at least, is this muscle type nicotinic receptor. It sits postsynaptically at the neuromuscular junction and mediates neuromuscular communication. In the absence of agonists, they predominantly adopt these resting conformations, which have low agonist binding affinity and a non-conductive channel pore. And upon acetylcholine release, they transiently adopt these open states that have a high agonist binding affinity and are conductive to cations. And then in the presence of prolonged agonist exposure, they adopt these desensitized states. And really the critical thing that I'm going to try and talk to you guys about today is the importance of understanding these conformational transitions between these various functional states. And I want to do that by first introducing the effect of altering these kinetics in vivo. So what I've shown over here on the left is a single channel recording of a wild type nicotinic acetylcholine receptor. And on the bottom, I've shown two mutations that come from patients with a disease called congenital myasthenic syndrome. And so in the first mutation here, we have what we call a slow channel syndrome, where the channel is much slower to close, and so it stays open for a longer period of time. And on the bottom, I've got a fast channel syndrome, which is much faster to close and so is open for uh, shorter periods of time. And what's important about that is how it manifests in the patient. So over here is a miniature end plate current. So this is a recording from the skeletal muscle of a patient upon the release of a single quant of acetylcholine. So you get an influx of cations and then this decay back to baseline, which allows you to rapidly fire and then ultimately cause subsequent muscle contractions. And so if you have a slow channel mutation, you actually prolong this decay, and the opposite's true in a fast channel mutation. You make this much faster. And what's really critical about this is when we try to treat these mutations in patients, we either broadly inhibit or enhance neuromuscular communication, and that ultimately just changes the amplitude of this initial response. And for that reason, it has very limited effectiveness in patients. And so, if we want to understand how these mutations ultimately manifest, and if we want to treat these things and cure disease, we have to really understand the conformational states that are being adopted during these gating transitions, so we can develop things that act specifically on the mechanism of action. And so, people have been modeling the kinetics of acetylcholine receptor gating for almost half a century, and we have a pretty good idea that these receptors have to exist at least these many states. You have resting states that can bind either one or two agonist molecules. These resting states then have to transition into some sort of pre-open intermediate state before they can open with either one or two agonists bound. We currently have structural information on the resting state, which you can just capture in the absence of agonist, or you can capture structures in the presence of a saturating concentration of agonist and get these kind of open or desensitized states. But we have no information as to any of these, what any of these pre-open intermediates look like. And so the goal that we had in this project was, first of all, to try and figure out what these intermediate states might look like and figure out a way to actually capture them with cryo-EM. So before I go into kind of the efforts we've done to do this, I just want to briefly introduce the structure. This protein has three structural domains. There's this extracellular domain that contains the sites for agonist binding. There's a transmembrane domain that contains the channel pore. And then there's an intracellular domain, which isn't important for this talk. There are these five subunits that are positioned pseudosymmetrically around the central ion pore. Again, for this talk, it's really critical just to know that there are these two principal alpha subunits that form one side of the binding site, and then the two binding sites are formed at the interfaces between these principal alpha subunits in either a delta subunit at one side or a gamma subunit at the other side. And then all of these subunits come together in the transmembrane domain to line the channel pore. And so the consensus in the field for several decades has been that nicotinic receptor opening occurs via some sort of concerted allosteric mechanism due to its kind of roots in the MWC model of allostery. And to illustrate that, I've shown some of the work done by Tony Auerbach at the University of Buffalo, where they performed phi-value analysis on a ton of residues throughout the protein and saw that residues near the transmitter binding site moved earlier in the gating transition. And as you go down the channel, things start to move later and later in the gating transition. And so this was thought to tell us about the gating transition being this conformational wave. So binding in the extracellular domain would slowly come down the channel and then result in channel opening. And in fact, in the other homomeric, pentameric ligand-gated ion channels, this is the glycine receptor, you can actually capture states of the receptor where the channel first binds, the extracellular domain transitions, and then you get this dilation of the pore that causes conduction. And so this is kind of the consensus of what a pre-active intermediate state might look like: an activated extracellular domain, a closed transmembrane pore. So how do we go about getting that? Well, again, Radu was going to tell us about some of the time-resolved methods he's done. We actually wanted to use more of a thermodynamic equilibrium kind of based method. And the idea is just basically, again, we can solve structures of these resting states in the absence of agonist. We can stabilize these open or active states by saturating the receptor with agonists. But if we somehow found a point near the midpoint of this concentration response curve, we could theoretically drag these kind of energy wells for any intermediate states down a little bit and hopefully populate enough particles there that we can actually capture them by cryo-EM. And so in order to do this method, we actually first needed a dose response curve. And so of course, we were interested in getting this in a few different methods. The contemporary way to do this is by electrophysiology, and we did that in various systems, and we typically get a normal EC50 around 10 micromolar. However, what we found is using this ethidium bromide binding assay that was developed in John Benziger's lab, that when we reconstituted the receptor into lipid nanodiscs, we actually got a concentration response curve that was shifted to the left by about 2 orders of magnitude. And this wasn't actually the case using the same assay for native membranes. And so for some reason, we can talk about it in the question period, the nanodiscs seem to shift the response curve to the left. But this gave us a target to look at to start recording data sets at subsaturated concentrations. And so I went to France and I worked in Hugues' lab, and with the help of Lefteris Zarkatis, who did a lot of the electron microscopy with me, we were able to get structures of the receptor in the absence of agonists in this resting state, in the presence of agonists using a saturating concentration of acetylcholine. And then we collected 5 different data sets at various subsaturating concentrations of acetylcholine, processed them all the exact same way, and then took a look. And so what was immediately evident when we did this is obviously in the absence of agonists, we got this uncapped loop C and no density for the bound agonist. When we looked at saturating concentrations, this loop C would cap inwards around the agonist and you would get density. But for all of these subsaturating concentrations, we would get very heterogeneous density in the binding sites, which was exactly what we were looking for. And this is very indicative of there being a conformational ensemble. So many different conformations that are being averaged around this binding site. And so I went through, I processed all of these things using various sophisticated conformational selection algorithms for cryo-EM, was able to pick out a monoliganded state by taking almost 2.5 million particles and processing this down into a small subset of about 130,000 particles that represented this monoliganded intermediate state. And so we can talk about that again in the questions if you guys want to hear more about how I did that computationally. But for the rest of the talk, I'll just kind of talk about what we found in these structures. So we're left with this unligated state, a monoligated state where the alpha gamma loop C has transitioned to a capped conformation with agonist bound, and then a diligated state with both sites bound. And so starting from the top of the protein in the extracellular domain, we can see that at the alpha gamma site that has the agonist, the loop C has completely capped into an almost completely active-like conformation, while the alpha delta site remains completely unactivated. And if we just look at a morph through those three states, you can see there's the initial binding of the first site, and then a subsequent binding of the second site around the second agonist. Pretty straightforward. Where things got a little more interesting and a little less straightforward was when we started going down the channel. And so this is at the interface between the two domains, the extracellular and transmembrane domain, where these two domains are thought to communicate. And we know that when this protein activates, this proline at the top of the pore lining M2 helix has to go past this valine residue. And in the alpha gamma site where the agonist is bound, this proline has almost fully transitioned to an active-like conformation. But in the alpha delta site where there is no agonist bound, this proline has already started to adopt a more active-like conformation despite no agonist being bound there. And we can kind of view this on the left. If we look at the alpha gamma interface first, you see initially a movement of loop C and a concurrent motion of this proline away from the pore, and then not much motion as we go from the monoligand to the diligated state. In the alpha delta interface, on the other hand, what you see is no motion in the binding site, but this subtle priming of this M2, M3 interface loop before you get full activation. And so that manifests itself in the pore as this asymmetric kind of opens closed state where the alpha delta helix has not transitioned whatsoever, and the alpha gamma helix has gone to an open-like conformation. And so we get an intermediate sized pore, which we showed by molecular dynamics simulations to be closed. And as you can see from this morph, you get an initial movement of this alpha gamma helix. And then when the second binding event happens, you get this full dilation of the pore. But if we look at this again, you can actually see this priming movement happening before the second binding site is happening. And so what we believe is happening now based on these structures is instead of this concerted allosteric transition where things move in the extracellular domain and then come down to the pore, what we actually see is binding to one site transitions part of the pore, and that helps prime the second subunit for a next binding event and helps explain some of the cooperativity that we see in these proteins, which is really interesting. And so we can see that again here with just the two domains overlapped. You get the binding of the one subunit and the subsequent movement of the M2 helix away before the second binding site comes and fully activates the protein. Now, the last thing I'm going to show before we get to the questions is to get back to where I started this talk and just the importance of understanding these allosteric transitions and the different states that underlie channel gating. And I think it goes back to this quote by David Calhoun, who said that kinetic models used to fit single channel data are useful only insofar they are sufficiently good approximation of the actual physical reality of the receptor's conformations. And so this is to say that we can kinetically fit any number of states to single channel data, but unless they're grounded in reality, it doesn't really matter what we put there. And so we turn to some single channel electrophysiological recordings from Corey DaCosta's lab, and they looked at these over a broad range of acetylcholine receptor concentrations and then fit them to a number of kinetic schemes. And what they found is that if they constrained these single channel recordings to work through a model where the receptor could bind agonist, adopt this pre-active intermediate primed state, and then could open via a single ligated or diligated state, that they got better kinetic fits to this model, even though it had fewer number of variables than the other models that they were testing. And so it seems to fit the electrophysiological data better than anything we've had before, and it seems to be grounded in reality. And so we think that we've found kind of one of the key parts of how the kinetics of channel gating work. And so with that, I just want to thank everyone that contributed to this. So obviously, John Benzinger, where I was doing my PhD, and some of the people in that lab. Corey DaCosta was also at the University of Ottawa. They did the single channel recordings. In France, I went over to Hugues Noury's lab in Left Terrace, spent many weekends and holidays with me on the microscope collecting all this data. So I got to thank him and Francois' lab for doing the molecular dynamic simulations. And then my new lab here, the Brohan lab at the University of California, Berkeley, for all of their support. So I'm happy to take any questions.
Thank you, Mackenzie, right on time. And that was a very fascinating talk, great research. If we have questions for him, please could you drop it on the chat box? While we wait for others to ask you questions, Michael, please, can you unmute and ask him your question?
Great talk, and I suppose you could probably anticipate my question. Wondering about
Let's
lipids
go
and
back
maybe.
to the nanodiscs.
Yeah,
Go
I
back.
know, but you did say you might mention it, so I thought I would ask.
Yeah, no. I think it's really it's one of the things that we still don't understand, which is a bit unsatisfying of an answer. But I love to talk about it because I think it's really interesting. To be quite frank with you, when we were doing this project, I did these assays of the reconstituted sample in nanodiscs. So we just detergent solubilize, put into MSPs, just a very standard process. This ethidium bromide binding assay is um, it's a conformational selective tube. You've all used ethidium bromide, probably. Well, maybe some of the older audiences use ethidium bromide for DNA gels, but it's a nice conformational selective probe for the nicotinic receptor. It only binds when it's active, so we can use it in a similar way as these electrophysiological recordings. And so I was pretty shocked to see how far the curve had shifted in the discs. And it wasn't until we got into the reviewer comments that they asked us to do this on the actual native membranes. And for the most part, I just assumed that this was something to do with the assay itself. It wasn't anything to do with the disc. But when we did the actual assay on the native membranes and found it was very similar to the electrophysiological recording, it kind of uh, it perked my interest as to exactly what the disc was doing to the protein's function. And I think that it highlights kind of I don't know, maybe one of the main themes of the conference, which is we want to get towards much more native environments for these proteins that are very sensitive to their lipid composition. I think that other types of discs would be very interesting to test via this, but I think ultimately what we really want to do is get towards full vesicle structures, cell intact structures, liposome structures, and eventually even some cryo-ET if we can get there. Because I think that removing all of these solubilizing agents would be the gold standard, although it's difficult to do at this time. But yeah, I'm interested. I'm interested too. We don't have a good answer for you. Sorry.
Yeah, I'm wondering if you have any hypotheses about lipids that might be critical for those conformational changes.
Yeah. Yeah, so we've done a lot of work on that. I know anionic lipids, particularly PA and cholesterol are really critical to the function of this protein. We made sure that the lipid environments that we use for the discs are all of the lipids that we need to activate the channel. And so I don't think it's something about the lipid composition per se. I think it's something about the physical properties within the lipid nanodisk. And so the ordering of the bilayer is supposed to be something that really influences this channel. As you could imagine, cholesterol and PA are both two lipids that influence the membrane order quite a bit. And so I think there's something about the compression of these disks that's ultimately limiting the conformational ensemble that we get. But that would be my best guess, and I would try to test that using some other system.